morning, good afternoon, everyone. We'll begin here in just a moment. Hello, everyone, and welcome to today's keynote webinar titled Delineating the Pleiotropic Impacts of Obesity on T-cell Function and Immunotherapy Efficacy. This webinar is part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now, before we begin, I would like to remind everyone that today's event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing the presentation, click on the Help Desk button down from the lobby within the event. And finally, as a reminder, today's presentation is educational, and it offers free continuing education credits. Click on the continuing education credits image located on the right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speaker, Dr. Curtis J. Henry, Deputy Associate Director of Diversity, Equity, Inclusion, and Access, the University of Colorado Cancer Center, Associate Professor of Immunology and Microbiology, the Margaret Turley Groen Chair in Translational Cancer Research. For a complete biography on our speaker, please find it on the left of your screen. Curtis, welcome. You may now begin your presentation.
So I know this is an international conference, so I'll start off by saying good morning, afternoon, or night to all of the conference attendees. And also before I begin, I want to thank the organizers of this conference for providing this platform for me to share our research, which I'm excited to do so with this community. I also want to give Christy a special thank you for that excellent introduction. And then also Melanie Chan for helping me with the logistics of actually getting this presentation set up. Now, I'm not trying to make any enemies out there. So I apologize for the very delicious looking foods that I'm showing you. But this ties into the focus of the day's talk, which is really trying to understand how our overindulgence in certain cuisines can impact our immune system's ability to fight cancer, and then also the efficacy of the emerging classes of immunotherapies that we're using to treat various forms of cancer. But before I begin, I think it's critical for me to thank the people who've done the work. I've recently relocated to the University of Colorado Anschutz Medical Campus. However, prior to taking this position, I was an assistant professor of pediatrics at Emory University School of Medicine, also associated with the Aflac Cancer and Blood Disorder Center of the Children's Healthcare of Atlanta, and then also the NCI-designated Winship Cancer Institute. And during my time at Emory, I had the privilege of training and working with these outstanding young scientists, as you see here today. And the work that I'm going to share with you has been primarily generated by Dr. Anthony Ross, who's on the right-hand corner and at the top of the screen. But Anthony was greatly assisted by everyone that you see on the screen. So again, without these individuals, the data that I'm showing you today wouldn't be possible. So thank you to all of these very, very special people. And so the overarching research goal of my lab is to understand how comorbidities which impact cancer progression, such as obesity, which will be a topic of discussion today, but we also study aging, how these comorbidities actually alter responses to therapies. And specifically in my lab, we focus on two classes of therapies, chemotherapies and immunotherapies, with the latter being the focus of discussion today. And so the reason why we actually are so focused on obesity is due to the following reasons. And this is data from the Center for Disease Control, which shows the United States and the percentage of adults in each state that presents with obesity, with the darker colors highlighting states where the percentage of adults that present with obesity actually exceeds 30% or more. And what you can clearly ascertain from this graph is that this is actually a national problem, where in most cases, in most states, the obesity rate actually is pretty high among adult populations. And as an African American, I'm also keenly aware that the obesity percent of adults who present with obesity are actually higher in African American communities, as shown by this graph. But this is also the case for Hispanic American communities, as well as indigenous American communities. Again, showing that this is a problem for all Americans, but it's also more problematic in underrepresented communities. Now, we've been able to track obesity or rates nationally for many decades. And so many scientists have started to use this data to develop mathematical modeling approaches to determine our projections in the next few decades. And this is just one of these predictions taken from this publication by Finkelstein and others that suggested in the next 7 years, over half of the US population will present with obesity, and that's specifically the adult population. So again, it highlights that this is a major problem now and will continue to be an issue in the future. Now, as someone who has studied pediatric cancers, I just want to highlight that this is not just an adult issue, as shown here by this the top navy blue line, but if you look at the green line, which shows the percent of youth that are presenting with obesity, and youth is actually defined children and adolescents between the ages of 2 and 19 years of age, as seen at the top of the graph. Looking at this particular rate, you can clearly see that the slope of the line actually shows that this rate is increasing among children and adolescents as well. And just to put some more current data to this particular point, in 2021, roughly 20% of the pediatric population in the United States presented with obesity. And just to put some numbers with this percentage, that actually represented 14.4 million children and adults that are afflicted by this state. And so as a person who studies pediatric leukemias, one of the things that my lab began to ask was, how does obesity impact survival outcomes in pediatric patients with specific subclasses of these blood cancers? And specifically, we were looking at this relationship as it pertains to B-cell acute lymphoblastic leukemia, as well as acute myeloid leukemia. And so this first graph I'm going to show you is some data that was generated a little over a decade ago by Sung et al., which looks at the survival outcomes in pediatric patients with B-cell acute lymphoblastic leukemia, or BALL, and they bin the data based on patients who presented as non-obese, which is that top line, and patients who presented with obesity, which is the checkered line at the bottom. And as you can clearly see, these authors noted a significant decrease in the survival outcomes of pediatric patients with BALL if they presented with obesity. Now, in the same year, another study produced by Naba et al., which looked at the same relationship, but for a different subtype of leukemia, acute myeloid leukemia, demonstrated roughly the same thing. Now, AML is much harder to treat in pediatric populations compared to BALL, and we can actually see that by that top dotted line, showing that the healthy weight patients, even with our best treatment options, only 70% of these patients actually survive over long periods of time. But similar to what the Sung et al. Paper demonstrated, and Naba et al. Actually showed that if pediatric patients present with obesity and this particular leukemia subtype, their survival rates were significantly reduced. And if we were to aggregate the data that were produced by several other groups, really showing the same effect, overall, what these studies collectively showed was that obesity actually decreases the survival rates for pediatric patients with leukemia, at least these two subtypes of leukemia, by at least 30%, which in our mind is pretty significant. And so with this data showing that obesity actually can reduce the survival rates, specifically in patients with pediatric B-cell acute lymphoblastic leukemia, one of the questions that we began to ask was, does obesity actually impact the efficacy or safety of cancer-targeting immunotherapies, particularly that there's published data, at least in the infectious disease world, and particularly with vaccines, that shows that this altered immunological state actually reduces the efficacy of immune cells. And so one of the things that we also wanted to study this relationship was due to the fact that obesity has been shown to be associated with at least 13 different types of very aggressive cancers, as shown here on this cartoon, some being breast cancer and pancreatic and colon, as you can see, as well as ovarian. But then you would notice that there's only really one type of hematological cancer, multiple myeloma, which actually seems to be associated with obesity. So again, we wanted to really look at the relationship between obesity and blood cancers, given the previously published data that shows that at least for two subclasses of leukemia, that it was actually bad for survival outcomes. Now, we're not the only group, obviously, that's looking into this. This area of research is actually starting to gain a lot of momentum, particularly within the last 5 or 7 years. And there's one paper that was published that really kind of set the tone of the field by Wang et al. It was actually published in Nature Medicine in 2019. And in this particular study, these authors looked at the ability of obesity to impact the efficacy of immune checkpoint blockade therapy, specifically those targeting PD-1 in the context of melanoma, which is a solid cancer. And surprisingly, what these authors found was that in the context of obesity, you actually had a heightened response to PD-1 checkpoint blockade, and patients and mirroring models actually responded better. And given that this was one of the first seminal papers, it kind of set the tone for the field, where the prevailing thought was that obesity actually improves responses to immunotherapies. Now, several people have decided that we should study this a little bit further. And then here are some of the questions that actually remain regarding this particular study, as well as others, including, are these responses durable? Because as I previously mentioned, obesity is actually known to compromise immune responses. And so one question is, over time, will these patients continue to fare better? And this is kind of what the focus of the topic today would be. Does this beneficial effect apply to all cancers? Cancers are very heterogeneous, even in the same cancer that relapses in the same patient. And so we wanted to understand, in addition to melanoma studies or solid cancer studies, will obesity also alter the responses or improve or decrease the responses in the context of blood cancers, specifically leukemia? And then also, does obesity alter the extent of adverse events or toxicity profiles which presents in patients? So these are still some questions that remain to be answered. And hopefully by the end of today's talk, we can shed a little bit of light on this relationship as it pertains to blood cancers. And so one of the first conclusions that we made when we started to look into the relationship between obesity and immune responses is that obesity compromises T-cell responses, specifically in pediatric populations. And we derived this conclusion from the following experiment. At Emory University, we have access to a ton of biorepositories of human samples. And so I utilized our CCTDC biorepository, which collects samples from non-cancerous patients. And they have data to actually look at these relationships in pediatric patients who presented as lean or with obesity. And so in this case, we obtain peripheral blood mononuclear cells, or PBMCs. We purified CD8 and CD4 positive T cells. These T cells were stimulated in vitro for 3 days, and then we used flow cytometry to look at T cell activation, with the question being, if we were to take T cells out of healthy patients without any disease, healthy pediatric donors, would we see any functional differences? And so today I'm just going to show you, in the interest of time, our responses for these CD8 positive T cells, which are these cytotoxic T cells or these killer T cells. However, I want to comment that the results look exactly the same for our CD4 positive T cells, which are T helper cells. But again, for today's talk, in the interest of time, we're just going to focus on responses from CD8 positive T cell. And what we can see in this top row is that if you stimulate these killer T cells from healthy pediatric patients, what we notice is after 3 days of stimulation, about 30% of these cells produce this very potent cytokine called interferon gamma that's used to awaken your immune system. Now, if you were to stimulate these purified T cells from pediatric donors with obesity, we noticed that the percent of cells actually that can make this cytokine is significantly reduced. This was not just the case for interferon gamma. This was the case for another potent cytokine called TNF alpha. And so if you look at the stimulated column, what we can see is that the percent of cells that were stimulated for 3 days from pediatric donors who fell within the healthy weight range was pretty significant, and then also significantly decreased if these T cells were stimulated from pediatric donors with obesity. Now, when we looked at two effector molecules that are that participate in the lysis of infected cells, including cancer cells such as preferrin and granzyme, we didn't notice a difference in the percentage of cells that actually produce either preferrin or granzyme B, which is the molecule responsible for inducing apoptosis or cell death in target cells. And just to quantitate this for multiple donors, again, what we found in terms of cytokines on that top panel is a significant decrease not only in the percentage, but also in the mean fluorescent intensity, or MFI, which is the amount of cytokine which is produced on a per cell basis. So the ability to produce these global immune stimulating molecules was actually significantly attenuated by T cells if they were isolated from pediatric donors with obesity. Now, although we didn't notice a difference in the percent of cells that produce preferrin, if taken from pediatric donors with obesity, we noticed that the actual average production of preferrin was actually lower in CD8 positive T cells if they were taken from donors that presented with high adiposity, and this was not the case for granzyme B. So overall, we concluded from this data that even in the absence of disease, obesity does compromise both CD8 and CD4 positive T cell responses. And so given this information, we really started to ask the question, these are the patients or the donors that actually would be receiving CAR T cell therapy. So if these T cell defects actually present prior to the disease, if you engineer a CAR T cell in a patient with the disease, will that CAR work as effectively, and will the patient actually exhibit similar responses based on BMIs? And so Anthony Ross, as I mentioned, who actually did majority of these studies, actually obtained a position at Raleigh Children's Health. And during his transition, he actually was able to collaborate with and actually join the Pediatric Real World CAR Consortium, which is a group that actually looks at some of these responses in pediatric patients. However, this group hadn't looked at the relationship between obesity and these responses. So Anthony took up the mantle. He actually published this abstract last year in Blood, so feel free to read it. But I'll show you a few snippets of the data. And one of the things I want to highlight is this data actually is comprised of data from 15 institutions. And we've actually analyzed 168 patients, which to our knowledge actually represents the largest cohort that's looked at this relationship between CAR T cell therapy for the treatment of B-cell acute lymphoblastic and leukemia in terms of obesity. And so when Anthony looked at relapse-free survival, which is the time post-treatment and the reappearance of treatment-related symptoms or either detectable disease, what Anthony realized and found was that there's a significant decrease in the amount of time before the disease appears if our pediatric patients present with obesity, even after receiving CAR T cell therapy. In other words, in these patients, the disease appears to come back faster compared to lean patients that receive this type of immunotherapy treatment. And again, we also noticed a trend in decreased overall survival, although it didn't reach significance. But what this data overall showed was that having obesity and receiving CAR T cell therapy treatment for B-cell acute lymphoblastic leukemia does impact the efficacy of this therapy. And so going back to our overarching hypothesis, from all of the data, we basically are working under the assumption that obesity-induced immunosuppression can indeed compromise T-cell-based immunotherapies, particularly in the context of blood cancers. And so to look at the function of T cells in pediatric patients with obesity and leukemia, we decided to return to our protocol that I described earlier. However, the difference here is we're actually looking at T cell responses from patients at diagnosis. And our question was, do these T cells actually behave differently even prior to engineering these T cells into CAR T cells? And so when we looked at this, we actually found some very, very striking data. And so again, these are looking at strictly CD8 positive T cells at the time of diagnosis. So when the patient actually enters the clinic how are they T cells functioning? And so even in unstimulated samples, what we find is that there are a higher percentage of T cells that are actually engaging the disease, at least by interferon gamma production, in patients that present in the healthy weight range relative to those that present with obesity. Now, we wanted to stimulate these cells to see if they actually had an ability to actually increase their function. And what we found was very striking. In the healthy weight patients with this particular disease, we noticed that there were an increase in the percent of cells that actually could make interferon gamma. And this was not the case in pediatric patients that presented with BLL and actually presented with obesity as a comorbidity. These T cells were unable to respond to additional stimulation. This was also the case for another cytokine that we looked at, again, TNF alpha, one of these cytokines that can wake up your immune system. In the healthy weight patients, we noticed that even without stimulation, the CD8 positive T cells were engaged in battle. And if you were to stimulate them further, they could actually increase their production of TNF alpha. But again, the CD8 positive T cells isolated from pediatric patients with obesity and BLL were not producing this cytokine. And even if stimulated very hard with the TCR stimulant, they were unable to make this particular cytokine. And so unlike what we observed for the cytolytic mediators without disease in pediatric patients with obesity and BLL, we notice a significant defect in the production of these cytolytic mediators, both preferrin and granzyme B. And again, if you were to stimulate these cells, CD8 positive T cells fail to induce significant upregulations of both of these cytolytic mediators. And again, we notice this both at the population level as shown in the primary data, but then also at the per cell level, looking at the MFI, we notice again, significant decreases in both cytokine production and the production of these cytolytic mediators, which are required to lyse target cells by CD8 positive T cells. So overall, from these studies, we concluded that obesity actually exacerbate T cell dysfunction associated with having B-cell acute lymphoblastic leukemia. So essentially, it's a double whammy. And so given these observations, we wanted to now kind of dig deeper into the molecular regulation of what was happening to our T cells, both in the healthy pediatric patients as well as those with BLL, and try to understand how did obesity impact the molecular signatures in each of these scenarios. And so I was very fortunate to collaborate with a great friend and colleague, Dr. Carmela Haynes, who is an associate professor in the Department of Biomedical Engineering, who was performing the very significant research at Georgia Tech University as well as at Emory University. And so what Carmela and I decided to do was actually perform a gene expression profiling analysis using the Encounter panel, which basically allows us to look at a targeted set of genes, in this case 780 genes, that are responsible for potent T cell activation. And we also looked at this again in human samples from pediatric PBMCs. We included 5 samples from patients that did not have leukemia, and we bin them into lean and obese categories, and then those with leukemia, lean, and those presenting with obesity. And from these studies, what we found was pretty interesting. Looking at principal component analysis, which bins data based on overall gene expression profiles. But in this case, we decided to look at these 29 T cell genes that are associated with high T cell activation. What we found by looking at the black dots is the T cells taken from lean pediatric donors without obesity. They clustered well. Surprisingly, those with obesity actually had profiles that resembled patients with leukemia. And what we noticed was that at least with some of these genes, having leukemia, whether the patient was lean or obese, basically led to similar profiles. And so looking at this in a little bit more granular detail by specifically looking at specific genes, what we noticed was there were certain genes that seemed to be affected or impacted more by having obesity and leukemia. And some of these genes, actually many of these genes coded for components of the T cell receptor. For example, we saw a significant reduction in genes coding for the T cell receptor alpha chain, particularly in leukemia, patients with leukemia that presented with obesity in those last 3 columns. This was the same case for genes encoding for the CD3 delta chain, the CD3 epsilon chain. the T-cell receptor gamma-2 chain, and then also to complement our functional studies, we also noticed that in the context of leukemia and in patients that presented with obesity, those T cells actually had very, very low expression of genes coding for that cytolytic mediator preferrin. And so when we looked at this a little bit more granular, just to show you this in a different way, again, the data popped out again regarding that the TCR was significantly impacted in pediatric patients with obesity and leukemia, as shown here. Also, mediators downstream of the TCR, such as FIN, was significantly decreased in these patients, as well as CD4, which can amplify TCR signaling. And then, as I mentioned, some of the cytolytic mediators, at least genes that coded for these cytolytic mediators, were expressed at the lowest level in pediatric patients with leukemia that presented with BLL, really corroborating that functional data that I showed earlier. Now, given this TCR signature, we decided to take a very unique approach to look at this in more detail. And so luckily for me, being in Atlanta, we have a very strong BME department due to Emory and the great collaboration with Georgia Tech. One of the great scientists that actually conduct research there is Dr. Khaled Salida, and then the newly minted Dr. Anna Kellner. And so we decided to collaborate on a project to really look more at the TCR biomechanics when T cells were actually exposed to adipocyte secreted factors. And I'm going to show you that information now. Here's Khaled. As you can see, he has a very large group of very outstanding scientists, and that's Anna. And so one of the things that I want to comment on is when we think about T cell activation, we sometimes think about it in terms of a signaling pathway. But with this image, what I hope to highlight is that the first engagement of a T cell with any other cell, whether it's a malignant cell or a non-malignant cell, is through these biophysical interactions. And I think this video actually demonstrates that very nicely. What you're seeing here is an orange T cell that has found and ligated to a tumor cell. And what you can notice from this process is that this is a very dynamic interaction. This T cell is sampling multiple areas of this cancer cell, sampling for different antigens that will tell the T cell to either engage in killing or disassociate and then go look for something else. But this, again, is a biophysical interaction that basically governs what happens during this process. Now, using this information, Khaled developed what we're called DNA mechanoprobes, which is shown here in this cartoon, and I'll walk you through it. And so basically, you're looking at a technology, a bioengineering technology that basically has a DNA hairpin attached to a gold bead, which is attached to a glass cover slip. Now, the engineering part that's cool about this particular technology is you have a DNA hairpin loop, as you can see, that's jutting out in this cartoon. And we actually have used FRET, or fluorescent resonance energy transfer, to look at proximity information, such that when the fluorophore, which is indicated in red, is next to the quencher, which is indicated in black, no signal is given out. We can actually engineer the amount of force required to open up this DNA hairpin, and we can also engineer what's presented to the T cell. In this schemata, is showing you a streptavidin conjugated to a peptide MHC complex, so we could actually look at antigen specificity as it pertains to biomechanical interactions. Now, if a T cell successfully gauges its target, whether it's a peptide MHC or an antibody, or as I'll show you later in the presentation, an immunotherapy, if the T cell pulls with enough force to open up the DNA hairpin, what you will notice is that the fluorophore gets dislodged from the proximity of the quencher. In this case, you actually get a signal. And so using this technology overall, what we're able to see is we can get qualitative information, which basically means the number of signals that we can count, but we also can get quantitative information, because as the signal moves further and further away from the quencher, the signal gets brighter. So we're able to capture both pieces of information. And just to demonstrate how this looks, if you use microscopic approaches, if the video works, and if it doesn't, even if you look in this middle panel, what you can clearly see is some green specks, which basically represents a specific peptide MHC TCR interaction, with the green showing you actually where that contact is actually happening. And so we can actually measure this for individual interactions, and we can measure this again on a per T cell basis. And so using this technology, we are able to actually apply very important biological questions such as the impact of the adipocyte secretome, which I'll show you next, and really try to understand how these biophysical and biomechanical interactions both impact function, but then also potentially predicts the function in very, very real time and using accurate measurements. And so the question that we applied to this technology is shown here. So we wanted to ask if you actually exposed T cells to either unconditioned media, stromal cell conditioned media, or adipocyte conditioned media, would it alter the biomechanical forces that the TCR is able to exert on its target? Now, we decided to basically culture T cells in these three medians for approximately 24 hours. And the reason why we did this is because we wanted to recapitulate what would happen in a real world scenario. In other words, T cells would be bathed in these microenvironments before actually seeing their cognate antigen or their target. And so we wanted to see with this preconditioning, did it alter that first initial biomechanical response? And so we decided to do this first using murine T cells. And so again, just kind of hearkening back on the power of this technology, if you can see from the schemata and compared to a peptide MHC complex being presented to the T cell, in this case, we actually use an antibody that targeted CD3, which is actually expressed on all CD8 positive T cells as well as CD4 positive T cells. So you can ascertain this information in using polyclonal T cells as shown here. And so when we did this experiment, exposing these purified CD8 positive T cells first to unconditioned media or to adipocyte conditioned media for 24 hours, and then placing these T cells on our biomechanical probes, what we notice is, again, we get this nice bright signal showing that the TCR is nicely engaging this antibody probe and is capable of at least exerting 4.7 piconewtons of force to open these probes. Now, if these T cells were bathed in adipocyte secreted factors for 24 hours and then exposed to these tension probes, what you can clearly see is that there's an attenuation of the signal. And then if you look quantitatively, with every dot representing the average intensity for a T cell, we see a significant decrease in the amount of biomechanical forces exerted by the TCR, even when it's in contact with this very, very strong, high affinity anti-CD3 antibody. So this was one of our first demonstrations that the soluble microenvironment indeed alters the biomechanical forces that a TCR could exhibit on its target. And so with that, we also hypothesized that we could use this biomechanical information as a biomechanical biomarker, which could predict T cell function. And so the data I'm going to show you and for the rest of this talk suggests that this may be the case. And so the first immunotherapy that we decided to study was blinatumomab, which is also known as Blincyto. And so this is an FDA-approved immunotherapy that is used to treat B-cell malignancies, specifically those presenting CD19, as shown here in the cartoon. And so the way this bispecific T-cell engager is also known as a BiTE works is you basically have a portion of this immunotherapy that targets CD3, as you can see here. And when you ligate the CD3, this actually activates the T cell. So it activates the effective functions like proliferation, cytokine production. And if this is a CD8, cytolysis or the ability to kill the target cell is shown here by this pink cell. But also it actually brings these activated T cells in close proximity with the CD19 expressing cells, essentially serving two functions, one as an anchor and then also as a immunostimulatory modulatory agent to T cells. And so again, this is FDA approved to treat B-cell malignancies such as BALL, for example. And so what we decided to do was to now switch to a human system and to determine if human cells were actually impacted by the soluble microenvironment. But the way we decided to do this was to use a novel iteration of our tension probes, where we basically made our DNA hairpin, as I described before. We actually attached recombinant human CD19 to our tension probes, and to the recombinant human CD19, we actually attached blinatumomab to that. So basically, this is being presented to T cells when we actually put them into this assay. Now, what we decided to also do was to expose our human T cells, as we did with the murine T cells, to various conditions prior to exposure to our what we're calling our blender probes. And when we do that, what we can see is very distinctive. In that first column, looking at human T cells exposed to our unconditioned media or just our standard healthy media that we're using in the lab, when you expose human T cells to unconditioned media for 24 hours, and then to our blender probes, we can see nice bright binding of the TCR to these blender probes, and you're getting a nice tight concentric ring, particularly around the plasma membrane. In our stromal cell conditioned media, where we tend to actually see stronger TCR biomechanical forces, we actually see that this translates to our human studies, and that when you expose human T cells to this particular soluble microenvironment and then to the blender probes, we actually get a highly uniform pulling capacity for our TCR against this particular immunotherapy. However, if you look at this response in the context of exposing human T cells to adipocyte conditioned media, what we find is that this signal is significantly attenuated. In this study, importantly, we also looked at these responses from multiple donors, as you can see in the quantitative data at the bottom. And what we found is, regardless of the human T cell donor, exposure to adipocyte secreted factors or the adipocyte secretome significantly reduces the biomechanical forces that human TCRs are able to experience when encountering this FDA-approved immunotherapy, blinatumomab. So one question we wanted to address was, does this biomechanical force actually predict overall function and the cytolytic capacity of these CD8 positive T cells when they encounter leukemia cells? And so the way that we did this experiment was actually shown here. Again, we're using a human system. We co-cultured human T cells with labeled human leukemia cells, so we can essentially watch their elimination in the presence and absence of this immunotherapy, again, blinatumomab. And really, again, the question is, does the initial biomechanical properties actually predict overall cytolysis or the cytolytic capability of these T cells when they encounter leukemia cells? And so I'm just reminding you of how blinatumomab works in using the cartoon on the left side of the screen. But getting to the quantitative data, what we observed after 24 hours of co-culture between human T cells and then human leukemia cells in the presence of this immunotherapy was that you indeed get some killing as expected. We noticed that the greatest amount of killing was actually when T cells were first exposed to the stromal cell conditioned media. And as a reminder, this is also the condition that gave us our greatest biomechanical force properties. And in the conditions that gave us our weakest biomechanical force properties, even at this early time point, you do see some killing when T cells were exposed to adipocyte conditioned media for 24 hours, but it's significantly less compared to when T cells were exposed to either unconditioned media or stromal cell conditioned media. And so we decided to look 24 hours later, so 48 hours total of this experiment, with the hypothesis that maybe the T cells are killing more slowly in the adipocyte conditioned media. So maybe it's a kinetics issue. So when we looked 48 hours later, we were actually surprised that it was not a kinetics issue. What we found is that human T cells that were co-cultured with human leukemia cells in the context of blinatumomab, we actually noticed increased killing if the T cells were first exposed to unconditioned media or stromal cell conditioned media. However, surprisingly, in the context of adipocyte conditioned media, even after 2 days of killing, T cells completely stopped killing or stopped recognizing the leukemia cells, which really, really surprised us and really, really demonstrated that these biomechanical forces actually do predict the cytolytic capacity of our T cells, even in the context of an FDA-approved immunotherapy like blinatumomab. Now, the one thing that we couldn't gauge from this experiment was what impact was being exerted on the T cell or the leukemia cell, given that adipocyte secreted factors were in this experiment. So we decided to turn our attention away from T cells, at least for a little bit, and look at the impact of the adipocyte secretome on human B leukemia cells. And when we looked at that, there's two things that really jumped out. And so we looked at one human BALL cell line, which is called NOM6. In that top panel, we're looking at CD19, which is, again, the antigen that's being targeted by blinatumomab. It's also the antigen that's usually targeted in the context of CAR T cell therapy. And really, if you look at the column that says 48 hours, what we observed by looking at that dark red histogram is a downregulation of CD19 surface expression on these NOM6 cells after 2 days. We also looked at two immunotherapies, two immune checkpoint inhibitors that's expressed on the surface of leukemia cells, PDL1 and PDL2. Whereas we didn't observe any changes in the surface expression of PDL1, surprisingly, we actually observed that PDL2, which actually can suppress T-cell function, reduce T-cell proliferation, reduce cytolysis, it actually seems to be somewhat increased on the surface of these human BALL cell lines. And so to check to see if that was an artifact of this particular cell line, we conducted the same experiment on a different human BALL cell line, these RE8 cells. And again, if you focus on the 48-hour column, very similar to what we observed with these NOM6 cells, we observed a downregulation on the surface expression of CD19 and an increase in the immunosuppressive protein on the surface of the cell PDL2, suggesting that the adipocyte secretome is indeed reprogramming these human BALL cells to potentially reduce the amount of antigen that's being used as a target for these immunotherapies and to increase immunosuppressive mechanisms that would compromise the ability of a T cell to actually kill target cells. Now, with this particular data, we decided that this was worth looking at in our pediatric patient samples. And so I collaborated with a great friend and colleague, Dr. Sunil Riker. You can see his picture at the bottom right-hand corner of your screen. And we decided to look into bone marrow at CD19 levels on BLL blasts. And the reason why we decided to focus on the bone marrow is because this is basically the site of minimal residual disease, or MRD, and it's also the site of relapse. So when patients actually relapse, this is where the leukemia cells actually hide out, and this is actually where they are come back. And so what Sunil found from looking at our patient samples is that the amount of CD19 that's expressed on the surface of the cell is on surface of BALL cells is actually inversely proportional to the BMI. In other words, if a pediatric patient expressed high BMIs, we noticed that they also expressed the lowest levels of CD19 on the surface of their leukemia blasts that were found in the bone marrow, suggesting that this potentially could be an escape mechanism used by these cells in adipose-rich microenvironments. So to summarize what I discussed today, one of the things I want to really emphasize is that the obesity paradox may not be applicable to all cases of patients receiving cancer-directed immunotherapies. And I'll stop there to really emphasize that this needs to be looked at in a lot more detail. It really needs to be looked at in the context of the various classes of immunotherapies, and then also specifically in various disease contexts, and then also in patients that present with comorbidities like obesity or aging or other things that could alter immune homeostasis. In our studies, we found that T-cell responses are indeed compromised in pediatric donors with obesity, and that these these attenuations in T-cell responses are further exacerbated when you have the comorbidity obesity on top of the leukemia. Now, mechanistically, what we found that the adipocyte secretome significantly alters the TCR, both at the gene expression levels, but then also uniquely by altering the TCR biomechanical forces. And this in turn can actually negatively impact the efficacy of immunotherapies such as blinatumomab. And we are also working on CAR T-cell studies as we speak. And then overall, from a patient standpoint, by using the Pediatric Real World Consortium and then great work done by Dr. Anthony Ross, we were able to show that in pediatric patients receiving CAR T-cell therapy, that the BMI does significantly impact the response, with significant decreases observed in progression-free survival, suggesting that this will be an area that we'll have to contend with in pediatric patients moving forward. And so with that being said, I'd like to thank my collaborators at Emory University. Although I'm no longer there, I have great relationships with everyone that you see highlighted in that yellow box. I also was very well financially supported by Emory, also by Georgia Tech, and many of the foundations such as the American Cancer Society, the Mark Foundation for Cancer Research, and Swim Across America, as well as the Aflac Cancer and Blood Disorders Center. So I can't thank my colleagues and also the funding sources enough for supporting our research. As I mentioned, I have transitioned into several new roles at the University of Colorado Cancer Center, and at the University of Colorado Anschutz Medical Campus. I'm specifically housed in the Department of Immunology and Microbiology, and all of these different entities are supporting the continuation of this research, as you can see here in the columns on the right-hand side of your screen in white. If you need to get in contact with me, if you are interested in collaborations, I've included my current email address and my new email address at the bottom of the page. I'd like to thank all of you for your time and attention. And with that being said, hopefully we can open up the forum to some hopefully engaging questions that I am really, really excited to discuss with each of you. And so thank you again for your time and attention.
Curtis, thank you so much for your informative presentation. And as you mentioned, it is now time for our live Q&A portion of our webinar. As a reminder to our audience, if you have any questions for Curtis, please enter them into the Q&A box. Just type in your question and click submit, and we will answer all of your questions that we have time for. So Curtis, we're going to start with this question here. Does obesity alter the cholesterol content in T cells?
So that's a great question, Christy. Thanks for asking me that question. So yes, mechanistically, we did look at the ultrastructure of a T cell. What we found and what we are continuing to study in the lab are these changes. We noticed that cholesterol content actually increased in the T cell and actin levels actually decreased. So I actually have this really, really interesting analogy that I'm using for the T cells that are exposed to this adipose-rich environment. It's kind of like Pangea. So I'm hearkening back to a lot of our 7th grade junior high education for people out there. Remember, Pangea was the supercontinent before it all broke up into the continents, 7 continents that we know today. And so the ideal is when you have a T cell, you kind of need to form the supercontinent to concentrate the signaling to fully activate the T cells. And so by having more cholesterol, what you're having is now these very dense lipid domains on the surface of the T cells that can't come together because they're almost too heavy, and you don't have plate tectonics to actually move these cholesterol microdomains around on the T cell. And so we're really, really looking at how this change in cholesterol to actin ratio changed the activity of the T cell. So hopefully stay tuned. Hopefully we can get out some stories and some manuscripts in the next couple of years that really, really can show the impact of this increased cholesterol in the cell membrane of T cells when they're exposed to adipocytes.
What a great analogy. Thank you, Curtis. Let's go to this next question. Does obesity impact the extent of adverse events that arise after immunotherapy treatment?
Again, another excellent question. So for those of you in the audience who may not be familiar with at least CAR T-cell treatments, one of the main adverse events or toxicities that's associated with this type of treatment is called cytokine release syndrome, or CRS. In patients that also receive CAR T-cell therapy, there's also patients that present with neurotoxicity as well. So these are kind of the two main toxicity profiles that can result from these types of treatments. What we noticed from our study, which I didn't have a chance to show you today in the interest of time, was that we do see higher grade 3, grade 4 neurotoxicity profiles in pediatric patients with obesity receiving CAR T-cell treatment. We're still, again, mining that data, but our data does suggest that the toxicity profiles are or could be more aggressive in patients that present with obesity. And we think this is presumably due to the higher inflammatory responses that are already present in the patient prior to the disease, which gets exacerbated when they have the disease on top of the state.
Thank you, Curtis. Now we're almost out of time. Do you have any final comments for our audience?
I think the comments are, if you're in the research space, really try to make sure that the research you're conducting accurately reflects the human population and the human condition. Our main goals are always try to translate this research to the clinic. And I think we can more effectively do that by making sure that our experimental models actually reflect the people. Not just certain demographics, but all demographics. Again, being inclusive even in terms of how we design our experiments. So I think is going to be very, very important to being great stewards to the human race as we continue to work in this space.
Absolutely. Thank you, Curtis. And I do want to bring attention to your email at the bottom of the screen. As you mentioned before, if anyone has any additional questions or comments, they can always email you right there, your email address. But I do want to thank Thank you. And again, I do want to thank Dr. Curtis J. Henry for his time today and his very important research. Before we go, I do want to thank our audience also for joining us today and for their interesting questions. Now, any questions that we do not have time for today, and also those submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand for 2 years. That's through October 5th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Again, Curtis, thank you so much, and thank you everyone for joining us. Have a great day.
Thank you, everyone. Have a great one. Nice seeing everyone.